Please subscribe and hit the bell icon to never miss a video from Acuere Life Science Foundation. Myself, Dr. Shantanu R. Joshi, a clinician, a pharmacologist, and a drug researcher. Dear students, today I am going to tell you about GPCR. What is this? This is G protein coupled receptor. G protein is a type of protein. It is having 3 subunits which are coupled together, and that's why we call them G protein coupled receptors. This group of receptor is the most important group of receptor because many drugs, rather maximum drugs, maximum endogenous hormones work through these type of receptors. Now I am going to tell you something about the synonyms. What are the other names for this GPCR type of receptors? They are known as serpentine receptors. You can very easily see this is the snake-like structure, and that's why they are popularly known by the name serpentine receptors. They are also known by the name 7-pass receptors. As you can see, very easily one can note this point. This is the first pass, this is the second pass, third pass, fourth pass, fifth, sixth, and the seventh. And that's why they are called 7-pass receptors. One more name is there, that is, they are called metabotropic receptor. This metabotropic receptor, because they interfere in the metabolism, they bring about some chemical changes, and that's why they are known by the name metabotropic receptors. Now, the next thing is this GPCR family. This GPCR family is a very big family. Already I told you many drugs act through it. Here we are going to take the example of adrenergic receptors. These adrenergic receptors, all of you know, they are divided as alpha receptors and beta receptors. Alpha is subdivided into alpha 1, alpha 2. They are having again their subdivisions. Beta is subdivided into beta 1, beta 2, and beta 3. These all type of receptors are from the family of GPCR. This is one example I am telling you. There are many drugs. You can say even 70% of the drugs act through this type of receptor. This is not at all exaggeration. This is the fact. Now, my dear students, I am going to tell you the components of this GPCR. Now, this end is known as ligand binding domain of the receptor. This is known as effector domain of the receptor. This is that protein I am telling you repeatedly, that is the G protein. This G protein is having the 3 subunits, and already I talked to you about this 3 subunit. This is the alpha subunit, this is the beta subunit, and this is the gamma subunit. You will note that S is written there. This S is written here, S. S stands for stimulatory. There are GS, GI, GQ, and GO subtypes of GPCR. We are today considering this GS subtype of this receptor. These are transmembrane receptors. They are located on the cell surface. They are transmembrane receptors. Now, they act through this activation of, their activation depends upon this GDP, that is guanine diphosphate. I will explain you later how it works. Now, this is one more component which is not directly related with the receptor, but yes, the activation of adenylyl cyclase is the part of activation of this receptor, and that's why we call it effector protein. This is known as effector protein. This is the important part. These are, you can say, the components of this GPCR type of receptor. Actually, GPCR itself is having the word receptor. I should not tell you again repeatedly the word receptor. Okay, but that is not a big thing. Now, I am going to tell you how this work, how GPCR work, how it functions. And this is really a interesting part. Already I told you, my dear students, that this is the ligand binding domain, this is the effector domain. Once this drug, I am taking the example of adrenaline here. Once adrenaline, all of you know adrenaline works on the adrenergic receptors. I am taking the example of beta 1 adrenergic receptor, which are mainly concerned with the heart rate, and other activities are also there that I am going to talk to you. Now, when adrenaline like molecule is going to stimulate this receptor on one end, it is going to stimulate the receptor throughout its length, and ultimately this effector region, ultimately the effector region of the receptor will get activated. Activation of this end will stimulate this G protein, and stimulation of the G protein will bring about the change of attachment of GDP and removal of GTP. Actually, GDP means there are two molecules of phosphate and one molecule of phosphate is going to add. And this is how that GDP converts to GTP, and this additional phosphate molecule is going to stimulate this trimeric protein, and due to its high energy, it gets cleaved. It gets divided into alpha plus GTP and beta plus gamma as subunits. I am going to show you in the next one. This alpha plus GTP now will move to this adenylyl cyclase and will stimulate adenylyl cyclase. Now, in the next slide, I am going to show you the same thing. Now, you can see that a high energy molecule GTP plus alpha subunit are going to stimulate this adenylyl cyclase, and this beta and gamma subunits get separated, GDP is separated. Now this adenylyl cyclase get activated. Activation of adenylyl cyclase is responsible for conversion of ATP into cyclic AMP. Now this is a very important point. Rise in the cyclic AMP levels. Rise in the cyclic AMP level is responsible for the activation of pKa. What is this pKa? That is phosphokinase A. A is there because it is activated by cyclic AMP. For that AMP, A is being written there, or adenosine, and that's why A is written there. Now, do remember, pKa get phosphorylated. Addition of P is there, phosphate group is there, and this is how activation of phosphokinase A will take place. This phosphorylated or activated phosphokinase A is responsible for activation of many enzymes. This is very important point, because when the enzymes get phosphorylated, they get activated. Sometimes they get inhibited also, but maximum times they are activated. Phosphorylation means the enzyme get some change. It may activate, it may inhibit. Most of the time it get activated. And activation of those enzymes will give you the real effect of the adrenaline or the filling of this domain or activation of this receptor. All depends upon this enzyme, the enzyme which is activated. Many enzymes, it depends upon the cell. I am going to give you some examples, and for this adrenaline, this is the adrenaline molecule, and that will serve the purpose. Let us see. I am going to show you the animation first. This is the animation. You can very easily see that this adrenaline is going to stimulate. Then activation takes place. Then GTP binds. The activated portion will move to adenylyl cyclase. It activates the adenylyl cyclase. Now activation of adenylyl cyclase will convert ATP into cyclic AMP. This is how the cyclic AMP are going to increase in the blood, in the cell. Raised cyclic AMP will stimulate phosphokinase A. There is phosphorylation of phosphokinase A, and the phosphorylated phosphokinase A further will phosphorylate many enzymes. It depends upon which enzyme is phosphorylated and in which cell, and this is how the biological effect will be seen by activation of this GPCR. Now, I am going to tell you about the same. Now, I am taking the example of a cell which is there inside the liver, and that is what we call hepatocyte. Suppose there is a hepatocyte and adrenaline is going to stimulate this hepatocyte. The same thing happens. The alpha and the GTP will move to adenylyl cyclase, then conversion of ATP into cyclic AMP will happen, then it will stimulate phosphokinase A, and phosphokinase A will go for phosphorylation of the enzyme, that is glycogen phosphorylase. This glycogen phosphorylase get phosphorylated, activated, and because of that the glycogen in the hepatocyte is going to convert into glucose, and this is how the level of glucose in the blood by the liver is going to increase in the blood. The hepatocyte is going to deliver the glucose in the blood, and this is how this activation of hepatocyte by adrenaline, very important, is responsible for hyperglycemia. This is how hyperglycemia develops. This is one example I have given you, activation of one enzyme inside the hepatocyte. The second example is related with heart. In the second example, adrenaline is there going to act on the SA node, sinoatrial node. All of you know the sinoatrial node operates by calcium ion. Depolarization of the sinoatrial node depends upon this calcium. Now, I already told you the phosphorylated phosphokinase A is going to activate or phosphorylate many enzymes here. It is going to phosphorylate this pump, ion channel. Every ion channel is ultimately a protein. Activation of that ion channel depends upon its phosphorylation. The phosphokinase A, pKa, in the cell, by its activation by adrenaline, the whole process will take place, and last will be the pKa. pKa is going to phosphorylate this ion channel. Phosphorylation of this ion channel will allow more calcium to enter the cells of SA node, and this is how the depolarization of SA node will start. Automaticity of this SA node will increase, and this is how adrenaline will increase the heart rate by acting on the SA node. What exactly happens inside the SA node? That this is going to happen inside the SA node. This is one example of one cell. This is one cell of this SA node. This is how the second thing we learnt now, that adrenaline is responsible for increasing the heart rate. The third example is again from cardiomyocytes. I am going to tell you about the cardiomyocytes or myocardial cells. All of you know the myocardial cells of the ventricles, of the atrias. I am taking one cell out of it. My point is very simple. My point is that calcium is going to enter because of phosphorylation of this ion channel. That pK is phosphokinase A, that is the last product of that whole chain, is going to phosphorylate this ion channel. When the ion channel gets phosphorylated, there will be the entry of calcium ion inside the cell. And when the calcium is going to enter inside the cell, all of you will agree with me that intracellular calcium will increase. And rise in the intracellular calcium is responsible for activation of this actin-myosin coupling and actin-myosin coupling contraction chain. And this is how the contraction of the smooth muscle of the heart will take place, and it will result into, when all of them are going to contract, the force of contraction of the heart is going to increase. This is how adrenaline increases the force of contraction of the heart through GPCR. Now, this is the second point, this is the third point I told you. Now, the next thing is related with the patient of CCF. Do remember, my dear students, in case of CCF, congestive cardiac failure, where heart is not able to pump the blood at the requirement of the body because of weakening of the myocardial muscles, because of the old age, many times of the old age, many other reasons are there for the CCF, but most common is the old age. My dear student, when the heart muscles are not able to contract properly, naturally body secretes more adrenaline. It is the reaction of the body to the low pumping by the heart, and this is how we compensate the pumping by the heart, by increasing the secretion of adrenaline, catecholamines. And this adrenaline is going to contract the vessels, the blood coming to the heart will increase and will compensate that CCF. CCF, congestive cardiac failure, it is also known as congestive heart failure, the same words. This congestive cardiac failure or congestive heart failure is due to that weakening of the muscles, and is compensated by high level of catecholamines. Catecholamines is written as Ca. Now, temporarily you will get benefit out of it. But what happens when higher level of adrenaline is continuously there in the blood in that old age person? My dear students, there is phosphorylation by pKa. Already all of you know this pKa now. All of you know this pKa. Whenever adrenaline is there, the pKa is activated. This activated pKa is going to act on the mitogenes. It will phosphorylate the mitogenes. Mitogenes are the proteins. These activated mitogenes will move to the nucleus and will express some proteins. Expression of those proteins lead to the myocardial fibrosis. And this continuous stimulation leading to the myocardial fibrosis will end into the remodeling of the heart. The heart will become bulky. This effect of this adrenaline over the years is responsible for myocardial fibrosis and remodeling of the heart. Because of understanding of this process, the scientists were able to assume that if we are using beta blockers in the patients of CCF, if we are using the beta blockers in the patients of congestive heart failure, then it may reduce the chances of remodeling of the heart. Then experiments were planned, and the patients of the CCF were given metoprolol. And because of that use of the metoprolol over the years, the mortality in the patients of CCF significantly reduces. It reduces by 20%. Studies available. And this is how we today recommend use of beta blockers in the patients of CCF, because it certainly improves the survival capacity of the patients of CCF. Why I am telling you? This is we can propose the use of beta blockers because we understand the process, how it works, how adrenaline works. Now, the enzyme, we have seen the four things. The first was related to the BSL. Adrenaline increases the blood sugar level. Adrenaline increases the heart rate by acting on SA node. Adrenaline increases the force of contraction by acting on cardiomyocytes. and adrenaline in the patients of CCF can lead to myocardial fibrosis and remodeling. And if you want to prevent it, if you want to reduce that possibility, one should use beta blockers in the patients of CCF, and that is what I can say the clinical application of this understanding of how GPCR works. Thank you, dear students. Dear students, thanks for watching. Please like, share and subscribe.